Yeah, so let's get started. I think people can still join while I introduce the speakers. So again, my name is Abhishek Kognole. I'm an application scientist here at SilcsBio. SilcsBio provides a structure-based drug design platform and services, and we are happy to host this webinar from our collaborators at University of Maryland, Baltimore. So I'll leave the topic of this webinar to explain from the speakers, but let me introduce the speakers. So we have Dr. Anmol Kumar, who is a research associate at University of Maryland, Baltimore in School of Pharmacy in the MacKerell lab. And along with him, we have our co-speaker, Alex MacKerell himself. He's a Grollman Greek professor of pharmaceutical sciences at University of Maryland, Baltimore. And he's also director for Computer-Aided Drug Design Center at University of Maryland, Baltimore. And he's as well a co-founder of SilcsBio LLC. MacKerell Lab works on developing the core methodology and its applications, and SilcsBio team is often collaborating with the lab in bringing the tools to our commercially available platform. So, having said that, I would like to do a quick poll while people are still joining about how well you know SilCS technology. So if you can see the poll, you can go in and submit your responses. Okay. Yeah, we see a lot of new users who have never heard about it. So it will be interesting. We will have some short introduction to the SILCS method itself in this presentation. So thank you very much for that. And I will now pass the baton to Anmol, who can, the floor is yours, Anmol.
Yep. So first of all, thanks Abhishek for your introduction. I'm actually really happy to be presenting our novel method, SilCS-WATER, which accounts for the role of water in protein-ligand binding. Of course, water is crucial for the protein-ligand binding, and considering its impact, it is important to study it and reveal and study it in terms of drug design processes to make it better and more reliable. So in this talk, I will initially talk about the importance of water in ligand binding studies. Then I will talk about SILCS simulation, which is the core technology of SILCS suite of programs. Then after that, I will introduce Silcs-MC, which basically uses SILCS simulations for predicting the ligand pose and ligand binding affinity. And then I will talk about the development and application of SilCS-WATER, which is the main focus of this webinar. We will also apply SilCS-WATER to a protein ligand data set, and I will talk how SilCS-WATER improves upon Silcs-MC. So although we know this, but just to set the context, I will talk about the different role of water in protein ligand binding. So for example, there can be three common scenarios. One where water forms hydrogen bond network and supports or improves the interaction of the ligand and the protein. Another scenario can be, for example, there is an apolar group on the ligand, which forces the waters to rearrange themselves. And then there can be a scenario where, because of a large functional group on the ligand, the waters which were originally present in the apo form are eliminated. Now, depending on whether the changes in the enthalpic contribution or the entropic contribution, the free energy can change positively or negatively, impacting the free energy of binding of the ligand. So I will just show you an example where they conducted a experimental study where a single water was displaced, and that caused a 1,000-fold of difference in the experimental binding affinity. This work was published in 2019, where Darby and Darby et al. They conducted an experimental study to understand the thermodynamics of the ligand binding to influenza protein. So this, what you see here, is a influenza protein, and it binds to sialic acid and its derivatives. Along with sialic acid, it also binds with 12 water molecules in the active site. And these group of scientists, what they did basically was to mutate a residue which was alanine in the active site, which is not even in direct contact with the ligand. So they modified it to asparagine. And because of this functional group, the water which was present in the wild form gets eliminated. And it also causes the other waters to be perturbed, and which actually, so for example, you can see over here, the water 4, which is present when in the wild type, is eliminated in presence of asparagine. What this does is, I should use a little bit. Yeah. What this does is it reduces the binding affinity of 3 different uh, sialic acid derivatives, and it changes it by about 1,000 folds, which in fact means that the change in the binding affinity is not because of the ligands. It is actually because of the changes of the mutations that they have made from wild type to the mutated type. And if you see in terms of numbers, because of this release of water, the entropy is gained, and the gain in entropy is 8.6 kcal per mole. But there are other waters which are unhappy, and that causes enthalpy to be increased, and the increase is more than the entropic gain, and thus the free energy changes in a positive value of 3.2 kcal per mole, and effectively it reduces the binding affinity by about 1,000-fold. Now, just by, let's say, if we have a crystal structure, just by looking at the structure, we cannot determine whether the water is important or not important for ligand binding. So we need a method that can give us the energetics of how the water is interacting with the ligand. As I said before, SILCS is the core technology of the SILCS suite of programs, which is the basis for SILCS-MC and SILCS-WATER, which I will be talking later. But first about SILCS, it is a computational method for pre-computing the fragment molecular affinity patterns, where a variety of fragment molecules, which are essentially representative of drug-like molecules, and they have very different polarities. They are sampled along with the target biomolecule or the target protein in the presence of explicit water molecule uh, to run the MD simulation and to get the affinity patterns of these co-solute molecules. Now, the beauty of SILCS simulation is that it captures protein flexibility while representing the solvent and co-solute explicitly. And how it does it is over here. So it is, SILCS simulation is essentially a cycle of grand canonical Monte Carlo simulations and molecular dynamics simulations. So during the grand canonical Monte Carlo simulations, what happens is these probe molecules or these co-solute molecules are added or deleted based on a oscillating chemical potential such that a 0.25 molar concentration is maintained. And once we have a initial box, it runs through molecular dynamic simulation for 1 nanosecond. And since it is repeated 100 times, and we also have 10 replicas running in parallel, we effectively get a 1 microsecond trajectory of SILCS simulation. And once we have this SILCS simulation trajectory, we use it to obtain the affinity pattern of solute molecules. So for example, what we do is we bin the trajectories in 1 angstrom by 1 angstrom voxels. And depending on the probability of finding a solute in a given voxel, in the presence of protein and in absence of protein, it is first normalized and then Boltzmann transformed to get grid-free energy, which is essentially what we call as frag map. Now, upon completion, we will obtain various different kinds of frag maps. For example, the green isosurfaces that you see over here are apolar frag maps. The red ones are acceptor frag maps. The blue are donor. You can also see some light blue colored, which is indicative of positive frag maps, positively charged frag maps, and the orange represent negatively charged moieties. So what this information is about is that apolar functional groups of the drug-like molecule would like to bind in these regions of the protein, and so on and so forth. Another important factor point over here is that the water maps are also generated because water is competing with all the solutes during the entire SILCS simulation. Now, along with the frag map, we also get exclusion map. And exclusion map is this region that you see in mesh over here. So this exclusion map is essentially the region of a space, region of the simulation where no solute or the water ever sampled. And it is, in fact, the extent of protein flexibility during the entire SILCS simulation. And once we have both the frag map and exclusion map, we can use it for various different kinds of applications without the limitation of using the protein structure, so which is the important part. Now, currently, SILCS suite of programs has various different capabilities. For example, it can predict the binding site. It has pharmacophores. It has molecular docking studies. It has capabilities of database screening, protein-protein interaction, and biologics formulation, all of which are based on frag maps and exclusion map. Now, one of the primary uses of these frag maps is to compute the ligand binding affinities. And how it does it is the ligands are placed in the field of frag map and exclusion map, and no protein structure is required. And the ligand grid free binding affinity, which grid free energy, which is indicative of the binding affinity, is computed by first classifying the ligand atoms into the frag map types that I showed you before, and then summing up all the grid free energy for each of the atom to get the final ligand grid free energy. Now, for example, let's say this is the ligand over here, and if the aromatic nitrogen falls on to the imidazole frag map, if the aliphatic carbons fall on apolar frag map, the aliphatic NH and OH group falls on formamide and methanol oxygen frag map, and if the benzene falls on to the benzene frag map, wherever the coordinates are placed in the voxel, the grid free energy of that voxel will be used to get the final ligand grid free energy. Now, SilCS-MC is a molecular docking technique in SILCS, which basically utilizes these frag maps to find the optimal ligand binding pose and also the binding affinity. For how it does it is, let's say we have the ligand molecule. It is first converted into CGNFF atom types using CGNFF. CGNFF is also a part of SILCS suite of programs, which gives the topology of the ligand. And once we have the CGNFF atom types, it is then converted into SILCS atom types. In addition, we would need the solute frag maps over here. So what it does basically is that initially the ligand is placed in the binding site. So this placement can be within a simulation radius, which user will define. The placement can be user defined or it can be randomly placed. Initially, the ligand will be energy minimized, and then it will undergo Monte Carlo simulation sampling and simulated annealing Monte Carlo sampling. Within the Monte Carlo sampling and simulated annealing, the only difference is the temperature is constant in Monte Carlo simulations, and the temperature changes during the simulated annealing. And in each step of Monte Carlo or simulated annealing, the ligand will be modified either in terms of translation or rotation or changes in the torsional angle, and that will be a random choice. And the difference in the intramolecular energy of the ligand or the ligand LGFE will be used in the metropolis criteria to accept or reject the move. Now, for example, if we achieve the convergence, meaning let's say the user has defined that if we have 3 LGFE values which we get within 0.5 kcal per mole difference, then we say that, okay, the results are converged, and we finally get the binding pose and the corresponding LGFE value. Now, this brings me to the topic of today, which is SILCS-WATER. Now, it is a modification of SILCS-MC, where along with the ligand, which we now call as primary ligand, the waters are also being sampled. So, of course, both of these are converted into SILCS atom types. And over here, along with the frag map, the water frag maps are also used. So, as was the case with SILCS-MC, the ligand along with now the waters are placed in the binding pocket. And while there is a minimum distance that is maintained between ligand and water, so for example, 1.8 angstrom should be the minimum distance between the ligand and water which are being placed. The initial placement can be random, and then it undergoes energy minimization, intramolecular energy minimization. Then the MC and SA sampling continues. Now, there is a major difference between SILCS-MC and SILCS-WATER. During this MC sampling in SILCS-WATER, the ligands and waters are interacting with each other. And the random choice of how to make the changes is either one of the ligand or one of the waters is chosen to be either translated, rotated, or making changes to the torsional angle. And I don't know if you can see this clearly, but the metropolis criteria also is different. And for example, now not just the intramolecular energy of the ligand and the ligand LGFE, but also the intermolecular interaction energy of the water and the ligand and the LGFE of the water are used in the metropolis criteria. So if a SILCS-WATER simulation is converged, we will get the pose of the ligand and also the pose of the waters. And we also, of course, get the LGFE values corresponding to it. Now, as you can see that I have declared the ligand, the ligand which was used in SILCS-MC as well as primary, which makes the waters as secondary ligand. Now, there is no restriction that it needs to be just water. It can be any other ligand than water, which means that SILCS-WATER can be also used for studying allosteric sites. Now, to showcase our method, I am showing over here its results on 10 different protein targets, where we know that water is an important contributing factor to the ligand binding. What you see here is the binding site, the crystal structure of the binding site, where you see the ligand, crystal ligand, and crystal waters within 4 angstrom of the ligand. So these red spheres are the crystal waters, and the skeleton, green skeleton, is the ligand, crystal ligand. Now, you can also see that in different proteins, we have different number of waters which are near the ligand, and some of which are also hydrogen bonding with the ligand, making them essential for the ligand binding. Now, as a first step, we need to generate the frag map using the SILCS simulation for all these protein structures. And for generating 1 microsecond of SILCS simulation, it takes about a day on a GPU node. And that's the most expensive part of the entire protocol. Because once we have the SILCS frag maps, we can use it repetitively for whatever application we want to do. Now, right here, I want to emphasize about the correlation between the water frag maps that we have generated as a result of SILCS simulation and the crystal water positions. Now, I'm showing here 4 examples of the crystal structures that we have used. So what you see is a crystal structure with the crystal ligand and the crystal waters, which are in red spheres, overlaid with the water frag map that we obtain from SILCS simulation. In most of these cases, you will see that the water frag map is really close to the crystal water positions. You can see in all cases, this is the common thing. But of course, you see some waters, some crystal waters, which are not near water frag maps. And then you also see some water frag maps where there is no crystal water. And it is important to note that during SILCS simulation, we did not use the ligand, the crystal ligand, neither did we use the crystal waters for generating the frag maps. So essentially, water frag maps is giving us the water binding positions, but a rigorous comparison is not possible between the experimental location of the waters and how they are contributing to the energetics of the ligand binding. And this is where SilcsWATER comes in. SilcsWATER can give you the energetics. What you see here is the SilcsWATER output for 2 different proteins, the base protein and the CHK protein. Here are the number of crystal waters. So for example, in case of base, there are 7 crystal waters near the crystal near the ligand, and in case of CHK, there are 10 crystal waters. And as an output, you see the intertot E is the value that is the interaction of water with the ligand, and water LGFE value is the interaction of water with the protein. Water tot E is total of sum of intertot E and water LGFE. The way these calculations are done in SilcsWATER is that we start off with crystal structure of the ligand and crystal water positions, and we try to maintain the spatial relationship between these ligand and water. And for that, to make sure that we don't deviate from the initial position, what we do is we don't do any Monte Carlo simulation sampling. We only do simulated annealing sampling, only for 2000 steps. What we find is that the interaction energy between water and ligand can be both positive and negative, although you don't see a positive valued interaction energy over here, but we do see it in when we see more proteins. Which means that they can interact favorably with the ligand. The waters can interact favorably with the ligand or unfavorably. Same is the case with water LGFE. You see there are some positive values, which means that some of the waters are interacting unfavorably with the protein. But when we sum them up, we find the water total energy is always negative, which essentially is telling us that the water is favorably with the protein or it is interacting favorably with the ligand. So for example, if there is a case where it is interacting unfavorably with the protein, which is, for example, 0.18 kcal per mole of water LGFE means that it is interacting unfavorably with the protein, but it is being stabilized by a negative or favorable interaction with the ligand. And this gives us a very important information for the drug design, which is we need to understand that we don't have any a priori knowledge of the number of waters that are there in the binding site. So what we do is we, along with the ligand, along with the primary ligand, we are also sampling 15 water molecules. The reason why we are using 15 water molecules is because we find that in most cases, within 5 angstrom of the ligand, there is not more than 15 waters, which essentially means that user can change this default protocol. And our simulation radius is 7 angstrom. Now there are 3 different protocols that we can apply. We can sample the ligand locally and the waters locally, or we can, this is of no use, or we can sample the ligand local and water exhaustively, or the most general protocol, which is sampling both the ligand and the waters exhaustively. Now let us look at the input file for these different strategies. So for example, over here in local sampling of the ligand, as you can see, the variable called random init orient is false, which means that the ligand will be placed in the user defined coordinate, while it is true for secondary ligand, which is for us, it is water. So the waters will be placed exhaustively, randomly, and will be sampled exhaustively. Also, the amount of sampling which is allowed for, let's say, the translation, rotation, and torsional angle changes are more for exhaustive sampling, and they are less for local sampling. So that's the general input to give you an idea about how you will change. Okay, so as I said before, that we start off by using a arbitrary number of waters, which is 15. Now we have to find a way to deselect the irrelevant waters and select only the ones which are important. So our selection criteria is based on 3 different scenarios. One is that the water and ligand should be within a certain distance. Criteria for how the water LGFE values should be means how it interacts, how the water interacts with the protein, and how it interacts with the ligand. So we played around with 10 different criteria. So for example, the first criteria is the most lenient. For example, it will capture all the waters which are within 5 angstrom of the ligand. It will capture all the waters which are having LGFE value of less than 3. Now, just to give you a, a, a picture, the max LGFE value or the most unfavorable situation for a water can acquire a value of 3 kcal. So essentially, the criteria 1 means that it will select all the waters which are within 5 angstrom radius. And criteria 10 is the most strictest criteria. It will select only the waters which are within 4 angstrom radius. a 4 angstrom of the ligand, it will select the water LGFE value only if it is interacting favorably with the protein, and the water total E, which is the sum of interaction energy of the water with the ligand and the protein, should be less than minus 0.6. And the reason why we use minus 0.6 is because this is the energy which is available to the water at normal temperature. Now, once we have selected the waters based on our different criteria, now we have a new metric for LGFE or binding affinity in case of SilcsWATER, which is not just the ligand LGFE value, it is also the sum of all the water's LGFE values that are selected based on different criteria. Now, this is particularly different from SilCS-MC, where only the ligand LGFE is used as a metric for binding affinity prediction. Now let us see how our new metric of binding affinity prediction is performing based on different selection criteria. So as I said before, we have 10 different criteria, and the mean unsigned error and RMSE values that you are seeing here is based on the 10 different protein structures that I showed you before. So what was done is the ligand binding affinity is known, the experimental binding affinity is known. It is compared with the LGFE value that we get using different criteria, and then the unsigned error and RMSE is calculated. And we find that for criteria number 9, both these values are smallest, and they are smaller than if you just compare, if you just take the ligand LGFE, you don't take any waters, it is smaller than this. And more importantly, it is also smaller than the LGFE of which we obtain from SilcsMC. In terms of figure, you can see how well all the LGFE values are predicted. On the x-axis, we have experimental free energy of binding, and on the y-axis, we have the predicted from SilcsWATER with criteria C9 over here. And in these two cases, they are not so good. So the first one is SilcsMC result. This is the SilcsWATER ligand LGFE only, and over here is the LGFE that is calculated based on the ligand LGFE and the water LGFEs. And here is the result for exhaustive sampling of ligand and waters. It is the same case that the C9 criteria performs the best, which means that C9 is performing better than the ligand LGFE alone, both in SilcsMC and SilcsWATER, whether you do exhaustive sampling or local sampling. Now that was about energetics, but let us also look about how it does in terms of capturing the waters. Now again, we know how many crystal waters are present in each of those uh, protein structures that we used, and we find that the same criteria, C9, is performing the best, which means that the C9 is capturing the number of waters which are in good agreement with the number of crystal waters within 4 angstrom of the ligand. And of course, there is no water considered in SilcsMC, so you don't find that column over here. More explicitly, you can see that these are the proteins that we used, and these are the number of crystal waters in 4 angstrom of the ligand, and these are the number of silks waters that we have found. And they are generally in very good agreement. Now, when we look at whether the waters are, where are these waters? So we find that most of these waters are close to crystal binding poses, crystal water positions. And one of the most striking result is in case of cydatolone dehydratase, where there were 11 waters within 4 angstrom of the ligand. But out of those 11, 10 were rejected based on the criteria C9, and only one was selected, and the one which was selected is really close to the crystal water. So we see that our criteria not only helps us in rejecting the irrelevant waters, but it is also maximizing our chances of identifying the waters which are important for the binding. And to reiterate, it is that water distance should be less than 4 angstrom. The total energy of interaction of the water with the protein and ligand should be less than minus 0.6. And it also allows the LGFE value of water to be positive, which means that it allows those waters which are interacting unfavorably with the protein. Now, there is another advantage which we get because of the SilCS water sampling, and that is the ligand pose. We find that when we conduct the SilCS-MC sampling, exhaustive SilCS-MC sampling, and when we conduct SilCS water sampling, the ligand poses are better in case of SilCS water. Now, what you see in the first column is the RMSD, and how this RMSD is being calculated is by taking the atom to atom difference between the pose that we obtain after SilCS-MC and the crystal position of the ligand. So that is how we calculate RMSD. And for the COM, it is center of mass deviation, how the ligand pose is differing from the ligand center, the crystal ligand in terms of center of mass. And we find that in most cases, the ligand pose is better for SilCS water than SilCS-MC. And there is a reason for it. There is a physical reason for it. Why this happens is because in case of SilCS water, the ligand is being sampled in presence of water, which is in fact guiding these ligand in correct binding position. This is to say that in case of SilCS-MC, for example, the ligand may acquire a orientation which is probably originally occupied by water, but SilCS-MC is giving you a pose which is better in terms of or more favorable in terms of LGFE, but not necessarily in terms of ligand pose. Now, this brings me to the comparison of SilCS water, how it does in terms of large number of data set prediction. So what we did was, for example, for all these proteins, we already knew that we have more experimental binding affinities for more ligands. So for example, along with the crystal ligand, we know 44 different ligands binding affinities. Same is the case with other proteins, and which makes it a total of about 430 ligands. We predicted the binding affinity based on SilCS-MC and SilCS water. And these are different matrices, correlation matrices. So this is Pearson's R, this is predictive index, and this is percent correct. And we find that our SilCS water, the new method, is better in terms of 6 cases out of the 10. Of course, the MUE, which is mean unsigned error, and RMSE is not that great for all these cases. But if you see more carefully, for this case PTP1B, SilCS-MC itself is really well correlated with the experimental binding affinity. And in this other case, CHK1, the number of ligands is less, which makes the correlation criteria really sensitive, and the differences are not hard to catch. So the correlation metric is not improved in terms of numbers. Now, there is an important observation in this case. When we compare these metrics, these correlation metrics, with the ligand pose, what we find is that it is well correlated. So wherever we see an improvement, it is correlated with better binding pose finding ability of the SilCS water. So for example, In SilcsMC, for the case of base, the ligand RMST is 7.53, which changes to 7.28 lower in case of SilcsWATER, or same is the case with, let me show you a striking case. 4.95 was the RMST of the ligand for HSP90, which changed to 2.42, and you can see how well the correlation, the PR, PI, and PC have improved a lot. So essentially, it gives you an idea that whenever, if you know the crystal ligand position, and if you see that SilcsWATER is giving you a better RMST, it says that SilcsWATER will be better at predicting the binding affinity of any number of ligands for that given structure of the protein. Now, I think I have been able to convince you about the new capabilities of SilcsWATER. Now let us look at the computational time that SilcsWATER takes and how it compares with SilcsMC. We see that for SilcsMC, we conducted these experiments. We ran these Silks simulations on single core CPU, and of course, you can see that there are different timings. These are all in seconds. And on an average, the SilcsMC takes about half an hour for the complete convergence. Of course, it depends on the size of the ligand, because size of the ligand is being translated, rotated. So it depends on that. And when we see the timing of SilcsWATER, this is about 2.5 hours. Now, there is another fact that we used a stricter criteria for convergence in case of SilcsMC, which was that if 3 ligand LGFE values are within 0.05 kcal per mole, then we will say that SilcsMC is converged. But in case of SilcsWATER, we use more lenient criteria, which is 0.5 kcal, and which is why SilcsMC runs for more a number of MC and SA runs. So effectively, SilcsMC is even faster. But roughly speaking, SilcsWATER is 6 times slower than SilcsMC, but it is still much faster than other FEP methods which are available for determining the energetics of water. Now, both SilcsMC and SilcsWATER is also available on GPU, where we have seen remarkable speed up in terms of time. So as you can see, on an average, on a GPU, now the 2.5 hours has converted into 2.5 minutes. And for SilcsMC, it takes around a minute. Although it is not fair to compare just one ligand on GPU code, but it seems like the SilcsWATER is around 2 or 2.5 times slower than SilcsMC on GPU. Now, this brings me to the conclusion. So I will just highlight few takeaways from this webinar. So for example, the silk simulation itself, when we conduct a silk simulation, it is giving us a binding pose of the ligand. It is giving us the energetics, but we are not able to get exact values of how water maps are contributing in terms of a given specific crystal structure, protein, and crystal ligand and crystal waters. SilcsWATER helps in getting the energetics explicitly for each water that are involved. And our general methodology uses 1 ligand and 15 waters, which are being exhaustively sampled within a simulation radius of 7 angstrom. And we need to deselect those waters which are not important. And we do that by using a selection criteria. And this over here is the selection criteria. Now, if we compare the SilcsWATER with SilcsMC, there is a difference in the LGFE. LGFE is our binding affinity predictor. So there is a difference. In terms of SilcsMC, it is just the ligand LGFE, which acts as the binding affinity predictor. But in case of SilcsWATER, it is the sum of ligand LGFE and all the waters that are selected. when we see based on our selection criteria, the number of waters that we find is very much in consistency with the crystal waters, and it is not at a very high cost. It is just 6 times slower than SilcsMC. And also, if you compare with rigorous FEP methods, it is much, much faster. Um, and I hope that our new method will be able to give very reliable drug discovery process wherever the water is important factor. So finally, acknowledgements to Professor Alexander MacKerell, who supervised the whole development of SilcsWATER, Himanshu Goyal and Wenbo, who were integral part of the development, Mengxian, who did the GPU uh, coding of this SilcsWATER, and all the MacKerell lab members. I would also acknowledge CAD Center for the facilities, NIH for funding, and SilcsBio finally for making the code available to all of you guys. And the code, the whole methodology is now published in JCTC recently, which you can go and see for the more detailed information. Thank you.
Right. Thanks, Anmol, for that really nice presentation. The attendees can use the Q&A section to ask any questions if they have. I have received a few that I will start with. I think one of the questions Alex already answered in the chat. But another question that we have is, since the poses of the ligands in the SilcsMC and SilcsWATER are going to be different, how can you get the same number of ligands after reaching convergence? Suppose the
Sorry.
wants to say so the question is, since the poses of the ligands in the SilcsMC and SilcsWATER are going to be different, or are they going to be different? That's also maybe a question. And how can you get same number of waters? Maybe that's what they're asking after reaching the conversions.
No, remember.
Yeah, go ahead, Alex.
I mean, there's this one ligand, one primary ligand in both SilcsMC and SilcsWATER. SilcsWATER then includes additional explicit water molecules. We start with 15. And then in the end, with SilcsMC, we have just the one ligand plus the subset of waters that we select. So then when we're comparing Silks standard at SilcsMC, it's the LGFE for that ligand. Whereas with SilcsWATER, it's the LGFE for that ligand plus the LGFEs of the waters. So in both cases, we just have the same identical ligand. In SilcsMC, we also have the contribution of the waters. And that contribution of the waters also impacts the pose of the ligand.
Right.
Thank you. Another question is, my protein has different conformations. Can silk simulation capture large conformational change? I guess I can answer that, or Alex, you want to answer?
Yeah, I mean, the SilcsMC has local conformational flexibility within the order of like 1 to 1.5 angstroms. If you have two separate conformations, we suggest that you do individual silk simulations on each of those conformations. And remember that that silk simulation only has to be done once. So although that's more expensive on the order of, say, a day, several days on 10 GPUs, once you've got the silk simulation done in the frag maps, all the other calculations are very rapid, as the timings that Anmol showed. So we would suggest two separate silk simulations for two very diverse conformations.
Thank you. And other question again, I think this is pertaining again for silk simulation, is, can I use my own probe molecules for silk simulation instead of the default molecules?
Yeah, that is the core idea, that we can use any number of ligands I was just showcasing how it performs with the crystal ligand, but you can use any number of ligands as long as you have the frag map for
I
the
think it's a
target.
different question. Go ahead, Abhishek.
Yes, I think it's a different question. It's asking about the probe molecules used in the silk simulation. So by like along the validations that we have done along the years, we have come to these set of probes that work really well in representing different functional groups, and we have taken care about different inter-probe interactions that might not be really well converged in a regular simulation. So we can allow additional different probes, but then one will have to validate that different set of probes, in and how it's going to be used for scoring the ligands and everything. So, however, technically it is possible to mm, use your own probe molecules.
Right, and then also we also do have a set of halogen containing probe molecules. And then also, if you have different types of like maybe fragments, like which are sort of like monocyclic, bicyclic systems, the idea is that you take the SILCS frag maps and you would dock those into the frag maps to identify the positions of, say, your fragment molecules, if you're doing, say, fragment drug design, or we use that to help us to identify novel binding sites and so on.
And we have one more, which is, can SilCS-MC be used for the Drude polarizable model, and how is its performance compared to CGNFF?
Well, in principle, yes. And we have published a paper where we showed maybe slight improvements. It was interesting. We saw that with the frag maps from the Drude polarizable force field versus the additive force field, you had different, like more orientation effects and things like that. So there are differences between the two. Now, the problem is with the Drude polarizable force field, we don't have it running with Grand Canonical Monte Carlo. So we have to do additional sampling using MD and things like this. And at this point, those capabilities are not available from like SilcsBio. It's been work that we've been doing sort of in the MacKerell lab.
And I think there's one more. Can I use SilcsWATER or Silcs-MC in terms of PROTAC development?
Yes, you certainly can. I mean, we're sort of in the academic lab, we're sort of trying to improve the the use of SilcsMC for PROTAC development. But basically, you're allowing for ligands to interact with the individual moieties from the PROTAC to interact with the proteins, understand how they bind. With SilCS, we can also do protein-protein interactions, so you can predict interaction orientations between two proteins. So all that information can be combined for PROTAC drug design. And we are actively optimizing those capabilities right now in our lab.
Okay. All right. Looks like that's about it. Thank you very much, everyone, for attending the webinar, and thanks to both our speakers, Anmol and Alex, for presenting their research. And yeah, so if anybody has any other questions, they can. Oh, we got one more. I guess we have another few minutes. Are frag maps defined based on the size of the ligand? Have you come across cases where the water sampled sites are close to crystal water sites but not matching the energy criterion?
I mean, the water frag maps nicely recapitulate the position of waters in crystal structures. Now, which means favorable sites defined by the water frag maps are sites where these crystal waters, which means those are favorably bound. Now, one could with respect to the energy criteria, I think that that's something that we'd have to look in more. I'm not quite sure
No,
that.
I mean, to add to it, I would say that if there is a water frag map, the water LGFE values will be favorable for the crystal waters if they are present over there. But then there also will be some crystal waters which are present where there is no water frag map, but they are being held by the favorable interaction with the ligand.
Right. I think you have also a cutoff where a combination of the water LGFE and water interaction,
Yeah,
that can be
exactly.
added,
So
to.
the combination helps us to give an idea whether it is either interacting favorably with the ligand or with the protein.
Right. All right. Thank you. And yeah, once again, thanks everyone for attending. Please keep a lookout for notifications from SilcsBio. We'll be releasing a new version of the software suite in next few weeks. So thank you very much again. Have a happy
Thank
New
you.
Year.
Thanks a lot.
Thank you. Take care.